Hey, good afternoon and welcome. My name is Ryan, and I work with the ALS Association. Thank you for joining us for this ALS Association webinar. There are a few things we want for you to know before we begin. There will be time at the end of this webinar for questions. You should see on your screens an option for Q&A. You can type questions you have there at any time, and we will review them at the end of the webinar to provide answers. You can also send your questions in the chat box. We do have a very short survey that we are asking that everybody complete after the webinar. The survey will appear in your browser once the webinar ends, but it will also be emailed to everyone who attends today. This survey helps us to know your thoughts on this webinar and topics you would like us to consider in the future. Much of the feedback we've received from our community and in these surveys involves a desire for more available information about ALS research that is currently being conducted and how individuals can participate. As a part of the effort to bring that information to our community, we have a research team from Georgetown University here today to present on 3 ALS studies that they are conducting. They will be presenting on 2 studies that are a part of the Target ALS program, as well as an NIH-sponsored national study called Access for All in ALS Consortium, which is also known as All ALS. 4 members of the research team are joining us today. I'm going to introduce them all right now, and then I'll hand it over to them to discuss the studies. Afterwards, we'll do the Q&A, but again, feel free to send your questions in the chat box or in the Q&A box. So I'm going to start with Dr. Brent Harris, who will be presenting on both the Target ALS postmortem study and the All ALS study. Dr. Harris is a tenured academic neuropathologist and physician scientist. He investigates the pathogenesis and pathophysiology of several neurological diseases, including ALS, with the goal of discovering new mechanisms and targets for drug therapy. He is the director of neuropathology for the following organizations: MedStar Health, the Histopathology and Tissue Shared Resource at Georgetown, and for both the Georgetown Brain Bank and Pond Brain Bank at Georgetown. Joining Dr. Harris when he presents the All ALS study will be Dr. Shakti Nayar. Dr. Nayar is an associate professor of neurology, primarily focused on clinical care with additional components of academic work. She has served as a principal investigator on several clinical trials for ALS. She provides education to fellows in her role as Clinical Neurophysiology Fellowship Director. She is the director of an ALS Association multidisciplinary clinic and director of the neuromuscular division at MedStar Georgetown University Hospital. And again, she will be presenting on the All ALS study with Dr. Harris. The final 2 researchers I will be introducing will be presenting first. They will be presenting on the Target ALS biofluid study. First up is Dr. Nicholas Stryker. Dr. Stryker is an attending neurologist affiliated with MedStar Health System. He is fellowship trained in both sports neurology and neuromuscular medicine and works with patients for all neuromuscular diseases, including ALS. His clinical interest includes diagnosis and treatment of neuromuscular disorders such as ALS, and his research focuses on discovering new therapeutic targets for ALS and neuromuscular diseases. Dr. Stryker will be joined in his presentation today by Cassie Holmes. Cassie is a clinical research nurse practitioner passionate about neurology and rare diseases. After working as a research nurse practitioner with the NIH for 4 years, she returned to her alma mater, Georgetown University. She currently coordinates the Target ALS biofluid study and All ALS study startup activities. With that, I'm going to hand it off to the first presenters, Dr. Stryker and Cassie Holmes.
Thanks, Ryan.
Yep, thanks everyone for joining us today. Cassie, do you have the slides if you want to bring them up?
Yep.
Okay, so sorry, I'm having some video issues, so I'll just be on audio, but we'll be around for questions later. So I'm gonna start off. Yeah, thanks everyone for joining. So we're, part of the neuromuscular team at Georgetown. So we're really excited to be able to talk to everyone today and give everyone a sense of sort of what's out there and what we're doing. So one of the main studies we're going to talk about today is Target ALS, which is really a natural history study. For those of you out there who have familiarity or don't have familiarity with it, the idea is sometimes we're doing studies where we do an intervention where we're saying, okay, we're giving you this medication, and we're going to see how it changes things. And sometimes we say, okay, let's take a step back and really understand what's going on with the disease. And that's the type of study that this is, where we're looking for, really what is the underlying mechanisms behind ALS, so that we can try to lead to new biomarkers, earlier diagnosis, understand the genetics involved, and really create better care for ALS. That's really what we're trying to accomplish here.
Next slide.
So next slide. So, what is the scope of this? It's actually really big. So there are multiple institutions that are involved, and it's a growing area. So you can see within the United States, there are several centers on the East Coast, the West Coast, and the Midwest. There are several sites that have just recently gotten activated in South America, as well as in Israel. And also their partner sites in Asia that share and collaborate data with. So really the goal behind this, it's biofluid, meaning it's either things like CSF or blood or other fluids that we can use to analyze, that we're looking sort of longitudinally, meaning across a long period of time. And we're really trying to collect as much data as we can on patients, both in terms of the disease progression and also in terms of these biomarkers from the fluids. Right now, we're at about 14 clinical sites, diversity of populations, and in addition to that, we're right now at 134 patients, and it's growing. This is led by Dr. Bob Bowser, who's one of the lead PIs, and he's one of the major players in getting this up and running, as well as Brent Harris, who's on the call with us. Next slide. So, in terms of the natural history and trying to understand what happens, the way that the study is set up is the visits are timed approximately when people come in. So maybe in different clinics, people come in between 2 and 4 months, but there's a little bit of a window in which people can be seen, but approximately we have people come in every 4 months, where we do different questionnaires, different questions about how people are doing now, and we collect these biofluids, urine, blood, CSF, and that there's testing to be done at home in terms of speech, so that we can understand how those are changing. So, on the one hand, it can be a lot to do. On the other hand, we do try to time it with clinic visits. And although some of the things are more than we'd normally do, such as a lumbar punctures sometimes for certain patients are already standard of care. So it's a little bit more than what we normally do, but we try to make it as seamless as possible with the overall experience, including reimbursement for travel, so that we can gain knowledge about the disease without adding too much burden to our patients. Next slide. So what type of patients are we trying to enroll? So we're looking for healthy controls. Sometimes these are family members. So it's really important for us to know what normal is. And then for people who are affected with ALS, we ask about environmental exposures. We ask about people's health at every visit, and we try to collect samples of urine, blood, and CSF at every visit, knowing that it may not be possible to do these always. So although this is what the study would like, we understand that there are limitations sometimes in how often somebody wants or can get a lumbar puncture. And then in addition, there's the at-home testing, which I mentioned, for speech, which can also be done. The total length for the study is visits that's 12 months apart. So we're looking at a total period of time in which we're doing these sequential visits. So why is it so important that we have all of these visits? It's important because we want to collect as much data as possible for as many people as possible. So it really helps us to have as many diverse patients as possible, to have normal controls, such as from family members or friends, and really try to get as many people enrolled as possible. And it's a great study. We also understand it can be a burden on the patient to come in to get all these tests done. And we always want to balance that with trying to collect new information, which can be beneficial to the community, really taking into account any individual need. And so I think on that note, I was going to hand over the mic to Cassie just to get her thoughts and her experience having worked with other clinical trials at NIH and the efforts that she's really taken on in order to make this successful so that we can learn and understand the disease better.
Yeah, thanks, Dr. Stryker. Yeah, so for particularly at Georgetown, I have participants that do kind of it both ways. Some participants prefer to come in and do it all with their clinic visit because they'd rather just come in one day and do it all at once. Other participants like to do it separately, so they come in a separate day and do kind of have their target day, and then they have like their regular clinic day. And so I really kind of just do whatever works best for them. Um, and I think these kind of natural history studies are just really important for helping understand diseases like this, because this is like going to help streamline and really accelerate the research. And this is the kind of research that I was involved before at NIH too. And this is really where I saw a lot of, um, the kind of new, exciting discoveries kind of comes out of this research. Great. I think.
Yeah, I think that's a great way of putting it. I think if we have questions about ALS, we're happy to answer them, or target ALS. Any other slides or any points we should go over, Cassie?
These are our final slides. I think next we pass it off to, I think, Drs. Harris and Dr. Nair.
Okay,
Excellent.
great. And if anyone has more questions, we're happy to answer them later.
Yeah, we'll do all the questions at the end, and I'll start sharing my screen. Let's see here. Okay, hopefully you can see my screen. So my name's Brent Harris. I'm a neuropathologist and a longtime ALS researcher, probably for about the last 25, 30 years, both at Stanford and then up at Dartmouth, and now here at Georgetown for the last 14 years. Target ALS does have the hope that everybody lives with ALS, and we're working very hard as a research community to find discoveries that will slow down this terrible disease and maybe stop it. The reality is, though, that people do die with this disease, and there's a lot to learn from the tissues that can be obtained after death. And so as a neuropathologist and a scientist in ALS, I've been helping to lead a post-mortem program for the last 9 years with a number of other sites. And I'm going to talk about those today. So together with Bob Bowser, we've been running this program for a number of years, and here are the sites around the country. We thought that it was going to be important to have these distributed around the country to East Coast, West Coast, middle of the country. Actually, we just brought on University of Edinburgh because they have a very long-standing program of working with ALS patients and their families and collecting tissues. The goals are to collect clinical data, tissues, to process and understand and confirm the diagnoses of ALS, to bank these tissues so that when researchers have a specific research question, they can ask for certain kinds of tissue and data. We can provide it in a de-identified manner to those researchers so that they can ask the important questions and push forward the science. We collect central nervous system tissues and skeletal muscle. And also we have neurologic disease-free controlled individuals who donate also to our bank. Both are exceedingly important. Here's our Georgetown Brain Bank team. You can go on to the Georgetown website anytime, or just put in Google Georgetown Brain Bank and you'll find our website. Dr. Khan and myself are the two neuropathologists, and Carolyn Ward and Marina Selenica are our coordinators for the Target ALS postmortem program. We're looking at all areas of the brain. We know that there are certain areas of the brain and the spinal cord and muscles that get affected in this disease, but we're also looking at multiple other areas. One of the hallmarks of neurodegenerative disease is that there are sometimes overlapping diseases. And with some patients who have ALS, they can also have a disease called frontotemporal lobar degeneration. And so understanding that relationship between these diseases is very important. We stain the slides with all different kinds of stains and prepare a neuropathological report and have those stains available for investigators if they want to know something about the histopathology that's observed. One of the projects that we started just a couple of years ago was to bring neuropathology kind of to the 21st century and start digitizing all of our slides. Some of you may have heard of this protein that is involved in most cases of ALS, all of the sporadic cases of ALS, and many of the familial forms of ALS, TDP43. And this is what a motor neuron looks like, this large cell here. And then the surrounding cells around it are glial cells that are probably just as important in your nervous system as this big fellow. But you can see these brown structures throughout, and then little specks of brown structures in some of the smaller cells. And it's this protein that is aggregating and causing problems with the way, the normal biochemistry of the way your cells work. So we and many investigators are focused on this protein and understanding it. So we've been collecting cases with our other sites for the last couple of years. We were given a grant from the Chan Zuckerberg initiative, and we're staining with TDP43 and then scanning all of these slides, digitizing them, and learning ways of quantifying those aggregates, and then making those available to any researchers who would like to also study them. And then also comparing it with people who have frontotemporal lobar degeneration. This is just one example of some of the kind of research that can be done with samples. We maintain these samples. We are custodian of these precious gifts for a very long time, and we preserve them in slides or blocks of tissue or in liquid format in fixative. We can also freeze tissues. Some research for genomics, proteomics, transcriptomics, metabolomics, all these different omics sometimes require frozen tissue and sometimes require fixed tissues. So we have both. We utilize very advanced computer cloud sites to take the data from our postmortem core, from the biofluids core that Nick and Cassie just talked about. We have another core that we're not talking about today, the stem cell core, and we can utilize different modalities to store these and coordinate them and with the logistics and with the clinical data, so that a researcher could ask for any one of these samples, cells, fluid, or tissues, and then say, tell me about this patient and what was going on with them, and what are the genetics behind what's happening with them. They may not have a genetic form of ALS, but there may still be important genetics that are related to the disease. That information then is given to them without any identifiers, but then it allows them to ask some very profound, interesting questions. So to date, the bank currently holds about 1,000 cases in all of those sites of people with sporadic ALS, lots and lots of control folks, people with familial ALS, and some people with frontotemporal dementia. These are some of the genes that have been identified and that are important in some of the familial forms of ALS. What have we done with these tissues? I told you about our project that we're working on here at Georgetown, but there are lots of other projects going on. We think at Target ALS and as a consortium that it's important to get these both to academic investigators, but also to companies, because the companies are going to utilize these biofluids and tissues to, in a very high throughput, find drugs that are and targets that are very important. So we have academic industry partnerships where we have academic investigators, researchers at companies who are working together to try to utilize these resources and ask the right questions to make the right drugs and start the clinical trials that are so pivotal. And we've had many more requests just in the last few years, which is exceedingly encouraging, that the pace of research is going up and that the samples are really being used in a productive way. We send samples all over the world. So we send them all to Europe, to many different sites in the US and in Asia, because this is a worldwide disease. And so we are sending it to researchers around the world. The researchers do not have to pay for the use of the tissues. The foundation really covers that expense. And we have a peer review process to make sure that the research is good quality research, because the tissue samples are precious. So these are just some examples of recent publications that have utilized these. So this is not just being used and not reported. These samples are being used and then put into top-notch journals and peer-reviewed studies. So there are challenges facing brain banks. There are decreased autopsies and tissue donations. So we're seeking partnerships with other local organizations, medical examiners, transplant groups to work with them. There's decreased funding to brain banks these days, although not from Target ALS. They have the funders of Target ALS have thought that it's very important to put these cores together. And we're also in the process of getting federal funding, which we'll talk about in the last project. We are having trouble in some cases getting control tissues, but we're better coordinating for that as well. And it's important that we communicate what's in our banks, specifically to researchers at national meetings and to families, such as you and patients. So lots of thank yous to our supporters at Target ALS, to CZI for the research funding that they provided, to the New York Genome Center, who does the genomics of every case that we go through, whether it's biofluid or tissue, so that there is advanced genomic information known about what's happening in these tissues. To all of our site PIs and coordinators and institutional support staff, but most important, to our families and individuals who participate in ALS clinical trials and banking efforts. And that's my last study. And I'm going to transition to a new study that is starting around the country that maybe you have heard about, called All ALS. And I'll have Shakti, if you want to share your screen, and I'll just introduce it a little bit. So the government thought that they really like the Target ALS programs that we've been running and other programs that have been similar, but they've been limited in scope just based on funding. And so for meaningful research, ALSA and other ALS communities petitioned and lobbied to have more early biomarker studies done so that diagnoses can be made quicker and we can better understand this disease. So this was brought before Congress and then brought before President Biden, and not just ALS, but there are some other neurodegenerative diseases. And a very large amount of money was earmarked specifically for this kind of ALS study. And one of the primary goals that I'm sure Dr. Nair is going to talk about is to make sure that everybody has access in this country to participating in ALS research. And so as a very ambitious undertaking, we've been spending the last year setting up 35 different sites around the country and deciding what the best way and what the holes in research were to be able to allow patients to participate in these kinds of studies, get integrated into ALS clinics and ALS multidisciplinary programs. And our site, of the 35 sites, a few of them have been activated. We're hoping to be activated early in the fall, so just in the next couple of months. So with that, I'll let Cassie take it over and talk for a few minutes about the All ALS program.
Hi, everyone. So this is Shakti Nairam, one of the ALS clinicians in our ALS clinic, and welcome again. So this is a coverage of an upcoming trial, as Dr. Harris was just mentioning, the Access for All and ALS trial, All ALS. As some of you may know, there have been various entities that have been pushing forward clinical trials based in various locations around the country. Over the past several years, there's been more collaboration between these various entities such as NEALS, the Northeast ALS Consortium, at the ALS Therapy Development Institute, and others. And what's really exciting is that this is a very vibrant landscape. The upcoming All ALS opportunity is going to build on these existing resources and enhance a lot of collaboration between them. And as Dr. Harris was just mentioning, this was made possible by a recent enactment of the Act for ALS, Access to Critical Therapies for ALS Act. And that allows for the public and private partnership to advance the progress in rare neurodegenerative diseases. And so the NIH in this case is partnering with private entities, which allows for this advancement. And so the NINDS, which is the neurological entity under the NIH, has been providing funding to advance this study. The goals are to create a large, flexible research consortium platform that can be modified over the years, provide opportunities to all living in the US with ALS, and those who do not have ALS who want to also help advance our understanding of what is seen in healthy controls, and also what's seen in those who develop ALS. This will allow for longitudinal studies, longitudinal natural history and biomarker studies. So biomarkers, we use that term very liberally, but as some on the call may know, the way that we diagnose ALS is really as a diagnosis of exclusion, looking at the clinical picture, looking at the physical exam, sometimes doing electrodiagnostic studies such as an EMG test, electromyography studies, and using these clinical studies at our disposal. There's not really a lab test, a blood test, a urine test, spinal fluid test that we use in our clinical settings as part of the ALS diagnosis. But it would be helpful to better understand whether there's a role for those types of tests. And in order to know that, we need to do these studies. That also, so aside from there being potentially some diagnostic relevance, there may be therapeutic relevance to biomarkers as well. So we may be able to use lab studies to determine whether a particular therapy is effective, for example, or whether it is likely to be effective for a particular individual. And the only way to know those types of pieces of information is to do these natural history studies, biomarker studies to start. So we're trying to build a large openly shared portal, sort of repository of data, so that researchers now and in the future can start to make more progress in ALS. So there's a lot of various acronyms that go along with this. AMP-ALS is the Accelerating Medicines Partnership Program, the partnership between the NIH and the FDA, as well as some private companies. And all ALS fits into that public-private partnership to help develop new diagnostics and treatments for ALS. Within that, there are the entities that helped to develop this protocol, the Barrow Neurological Institute and the Healy Center at Massachusetts General Hospital, and various sites that are overseen by each of them. 19 sites under the Barrow and 15 sites under Massachusetts General Hospital. And then there is a biorepository for lab samples and data portals for collection of information from things like questionnaires and speech assessments. So as mentioned before, there's 35 clinical sites really spread all over the country and overseen by the Barrow and MGH, Massachusetts General Hospital, respectively, all listed here. The goal is to enroll participants quickly, enrolling ideally more than 2,000, and allowing for a diverse group of patients to be enrolled. Those in different locations of different races and ethnicities, of various socioeconomic status and education level. And really we want to reduce any barriers to enrolling and be able to coordinate between the different entities. This program that we're doing today is one of many steps we're taking it for outreach to make folks aware of what's available. And we are so pleased that the ALS Association is doing this today and helping partner to get everyone informed. This study is really decentralized. So it allows for participants in various locations. It's not very centered on one particular institution driving the study and seeks to share information. There is the ability for e-consent, remote consent in cases where that makes it more feasible for a patient. And the video conferences for some pieces of data, remote nursing visits for collection of certain samples as well. There are a number of advantages to the consortium design of this. It reduces, as I mentioned, some barriers to participating in research. And this also improves the speed with which data can be captured and with which biosamples can be obtained by the research community. As I mentioned before, there's a number of different measures, including speech assessments, some assessments of strength, including movements of the limbs, head movements, but all measures that are straightforward for participants, so that it's not necessarily required to be on site in some cases. There's flexibility under this consortium design as well, which allows for the protocols to change, for research protocols to be added more easily, which is not something that we've seen previously in many clinical trials. There are two initial study protocols under all ALS. One is called Assess, the other one's called Prevent. And essentially, these two trials are going to allow for symptomatic ALS participants, really under the ASS study, to do biomarker assessments and other study assessments, either on site or entirely remote. There is an arm of healthy controls under the ASS study as well. Those would be on-site assessments. And really, these are not very frequent. They're essentially every 4 months. Asymptomatic people who may harbor an ALS causative gene can enroll in the PREVENT study, which is also under this all ALS umbrella. And that has a hybrid approach, on-site visits annually and some remote visits in between. And so by doing these on-site or remote visits, we can collect longitudinal biofluids, including blood and variants of that, plasma or serum from blood. Spinal fluid collection is optional. We can collect longitudinal speech recordings at home and electronic patient-reported outcomes. And if people participate in one of these studies under all ALS, they are allowed to participate in other ALS clinical trials as well. Just one quick thing to sort of explain the electronic patient-reported outcomes. What we're referring to there are certain self-administered questionnaires, which can be administered through an electronic platform. And so 8 sites are currently active. Over 20 participants are enrolled to date. And our site is anticipated to be active very soon this fall. So very exciting initiative. And we're happy to take any questions.
Thank you to the presenters for putting on such a great presentation. It's so informative. We do have some questions here in the chat. And I think just to start, if you could clarify when you're referring to biomarkers, what exactly that refers to. One of the questions is at diagnosis of ALS, there was no mention of biomarkers, that it was not genetic. Is there a specific biomarker that would have been mentioned when they received their diagnosis? So I think maybe clarifying even what biomarkers, what that term means would be helpful.
We all probably can add a little bit to this, so I'll start off because it began with mine, and then Dr. Harris, Dr. Nye have something else to add. And the reason I open with that too is I think it's a great question because I think biomarkers is so broad. I mean, there's everything can be a biomarker, from the way you walk to an MRI scan to a blood test. So really, it's a very broad field. And you know, in ALS right now, one of the challenges, which is why it's a great question, and one reason why it may have not come up during a clinic visit, is because we don't have a biomarker that says for certain, like, if you're positive for this, you have the disease. The closest we probably come to are things like genetics. And that's, you know, one of the areas of ongoing interest. I think in the context of what we're looking at with Target ALS and with all ALS, we're looking at some particular blood biomarkers, so markers in the blood that signify that you have ALS, or in the CSF. One example which I'll give, and then we'll see if anyone else has anything else they'd like to add, is neurofilament light chain is a very popular one that happens when axons break down. So that's one that actually one of the drugs, tofersen for SOD1 positive ALS, that's what the approval was made on. So these are very important markers, and it's a great question because biomarkers can be really anything that are a marker of biologic activity. I'll see if Dr. Harris or Dr. Nair have anything else they'd like to add in, or Cassie as well.
Yeah, I'll just say really quickly, the NEALS organization did a webinar just last week on biomarkers that some of you probably caught, and presented some of the data of how you can measure things longitudinally. And I think Dr. Nair did a great job of explaining what biomarkers were, that that they were not just for the initial diagnosis, but if you're on an experimental drug, how do you know that it's working? You know, you can know from symptoms and things, but you can also know with the biology behind, for example, neurofilament. And so to be able to say that an experimental drug is working, let's continue to do it and use it. Seeing positive results on biomarkers might be really, really important. And I'll stop there.
Okay. And I'm going to try to get to these questions in the order they came through. So the first question we got to was, have you learned anything yet from the natural history study? Or what are some of the...
Yeah, so I would say that, you know, the biomarker natural history study for target ALS only began a couple of years ago, and they waited for about a year to accumulate enough samples to then be able to start giving it out to investigators. So we're right at the beginning of understanding what's happening. But there is data that's presented at the National Target ALS meeting every year talking about some of the changes that correlate maybe the voice parameters and the speech parameters with what they're seeing in the biofluid. So it's really, really interesting beginning with. The postmortem studies are, in a sense, not a longitudinal study. There's what's happening to the tissues at the end of life and at the end of disease. And so that's one time point. And we think that having the combination of both the biofluids and then the understanding of the tissues where you can glean much, much more information together is going to be really, really impactful.
Okay. The next question, I'm going to combine these. What is the eligibility criteria for the Target ALS trial and the All ALS? Or is there eligibility criteria that someone needs to meet?
Cassie, do you want to try to tackle that one?
Yeah, sure. I'm happy to talk about that. So for both studies, you need to be 18 years or older, be able to understand, are capable of giving informed consent, kind of understanding the purpose and the risks of the studies. If you're an ALS participant, you will need to have a diagnosis of ALS. If you are a healthy control, then usually the studies require that you don't have any like neurological disease. Um, I know the healthy controls in target ALS, you cannot have you can't be like a genetic carrier for like a known ALS variant, but that you would be eligible for the PREVENT study for all ALS because that's really what that study is looking at. And then I know for target ALS, we do a speech score from the ALS FRS R scale. So there does need to be kind of a specific like target you're hitting with speech because they do want to catch people who are like earlier in the disease process. And then for target as well, the ability to be able to undergo a lumbar puncture is important as well. So if there's kind of issues there, we would recommend probably it's not the best study for you. And then I know all ALS does have the whole virtual or remote options for people that can't necessarily come to clinic or travel to challenge. So that's kind of great for catching people that want to participate but maybe have like more significant barriers to getting.
I will
Thank
add
you.
one other thing too, because this came up. Ryan asked me, you know, if you have a diagnosis of PLS, primary lateral sclerosis, you are eligible for the target study. And we do think that it is important to have related diseases, even if they are not exactly the same as ALS in these banks, because people will ask research questions wanting to compare the different diseases. So PLS is eligible.
Thank you. And the next question is how to get enrolled, how to sign up for these studies, where they can go to participate.
So you can participate at Georgetown or any one of the sites that is enrolling patients. You know, my suggestion is always, as you're starting to think about wanting to participate in clinical trials, whether they're treatment or trials or more participation longitudinal trials, to look at clinicaltrials.gov and see who the contact people are. We can provide information of how to contact people here at Georgetown, but if there are other sites that are closer to you and more convenient, then consider those sites as well.
Okay, let's see here. Is there a specific gene or biomarker that's impacted by sporadic ALS where TDP-43 is identified? That's I know that's a specific question. I don't know if
It's a
you're
good
able to answer.
research question. We think that TDP-43 is behaving abnormally in all cases of sporadic ALS and in many cases of familial ALS. The one that doesn't seem to have aggregated TDP-43 are people with mutations in the SOD1 gene. We don't quite know why that is. So TDP-43 has gathered a lot of interest in the research community, both as a marker of disease, both in biofluids and in tissues, but also as a potential target to try to prevent that aggregation. It's a very important biomolecule that binds to other proteins and binds to nucleic acid, so it can have some regulatory activities and it can have some important protein activity.
And just to add on to that too, that was the target upstream of that for Ataxin-2, which was the ALS Spire trial, which was a failure, but that was the idea behind it. And on the notion of biomarkers too, we recently just had our neuromuscular study group meeting up in New York. And, you know, beyond just looking at even genes, they're looking at these complex, what are called proteomics, where they look at multiple proteins and try to identify a pattern that could lead to something like increased TDP-43. So the biomarkers are huge, and they're just becoming, you know, more and more complex to try to identify, you know, what could be driving these and potentially be targets in the long term.
Okay, and I'm going to go a little out of order because this seems to be a question going off of that. It was just how can you get your TDP-43 level tested?
So, you know, I do want to just, you know, make sure everyone's clear on where we stand with research. So from an ALS researcher perspective, we are so excited that in our various conferences and meetings, there is more to discuss. We have more scientific advancements and better understanding of some potential biomarkers, some better understanding of what happens in the motor neurons in ALS. But we are really at that level where, you know, we are starting from a research perspective to better understand what the steps are that result in the loss of a motor neuron. That is the initial step. We then need more scientific studies. We need more clinical studies where we can see what actually happens in particular patients, various patients of different ages, different rates of progression of ALS, different sites of onset of their weakness and weakness patterns, those with a familial form of ALS, those with sporadic ALS. And once we have more information at a really broad population level, we can then understand those initial sort of scientific pieces of information. So what you know, what we were just talking about and what Dr. Stryker and Dr. Harris were referring to is the latest information on components in cells that may be relevant to ALS pathology. But this has not translated into clinical care yet because we have not gotten enough information in the research space yet to be able to translate it into clinical care. So that's what Target ALS and all ALS are seeking to do.
Okay. Thank you. Okay, as there's a lot of questions, so bear with me as I'm keeping track of them. As far as remote participation, could this work be added to the Synapticure EAP on CNM AU8 With that, everything is remote except for the blood work, which is collected in the home. And that is in the Q&A if I'm mispronouncing any of that.
So, and I think this was a question, I may have missed some component of the question, but I think this was a question about whether it's possible to concurrently participate in one of the expanded access programs that's currently available for the CNMA-08. It is possible to concurrently participate in either of the expanded access programs that's currently available, either the one for CNMA-08 or the one for predopidine as well.
Okay. Can German institutes or mixed US and German participate in any of these studies and funding? Can PALS participate anywhere in Europe?
Well, for the target studies, they are every year they are considering new partners to expand the diversity of people participating, and that would be a question to talk with the foundation and decide. They have limited funds, but they have a lot of fundraising to do more and more sites. The same is true with the postmortem core. We decided to expand to the UK because they had such a strong background of collections and were good partners. I do know that European countries and Asian countries can participate in clinical research through NEALS, and that's always a possibility. But I can't answer specifically for one site, at another location. But we certainly can put folks in touch with folks at Target ALS. For the All ALS program, that's going to be limited to the United States because it's funded by NIH.
Thank you. Okay, if someone was diagnosed with sporadic ALS in 2011, would they be eligible for these studies?
Yes.
Okay. Going back to the postmortem study, is there a distance limit for how close you have to be to a site in order to participate?
Yeah, we've actually gotten more funding from Target ALS. We used to have a limit of 50 miles, and we realized that was just not enough to serve the distribution of folks, and so we can work with folks 200 miles away, and if we're not the closest center, then there may be another center that can assist. So yeah, just contact us and we'll help coordinate and discuss these things. We really like to talk with families and patients. And it's actually very important that we talk with you if you're thinking about making that, very precious gift, so we can talk about how that would be coordinated, because there is a time limitation, and we do need to collect the tissues very quickly. So having some plans and helping you to understand the logistics so that your loved one doesn't feel stressed a lot when the time comes, I think is important.
Perfect. And this one might be more for Dr. Nayar. For the All ALS, with it, you had mentioned that a part of that is remote. Does that mean you can participate even if not living near the sites that are identified? Or do you have to be, is there a relative distance that you must live from those sites to participate in the remote part of that study?
Yeah, so this is structured to be able to do remote visits for exactly that reason. If someone is at a distance from the sites that are available, they still can participate in this.
Yeah, for example, if there isn't a one of the 34 sites in the state that you're participating in, then the two coordinating sites, the Barrow Institute in Arizona or the Mass General Healy Center, will assign one of the 34 sites and work with that site. Will work with you even though you're in a different state. That's not a problem.
And you do that remotely.
Yep.
Great. And it looks like this is the last. Oh, nope, two questions. So one more question that clarifying a previous question. Could this project coordinate with Synaptic Cure to use their blood work as well. And if you're, I don't know if you're able to answer that, or if you're familiar with
I'll
what's.
just say one thing as it came to my mind. These things are always great. We all love efficiency. The world could be a much more efficient place, but sometimes I can just say, and maybe Dr. Harris or Dr. Narakasi have any more information, but sometimes the problem is the labs or the testing for, breathing. They're slightly different machines, and so they kind of need to, for a particular study, all use the same machine for consistency. So although the efficiency idea is great, I think at this time it really has to be done by a certified particular lab for the study. Although it's something great to think about for all studies, having more centralization, I think probably at the time it needs to be done with the, the plans that are currently set up for these studies.
Thank you. And right now, this is the last question. It says, it appears that for those losing their functional capacity with a high progression for those with sporadic ALS, would not have a particular clinical trial, but the all ALS would change this in the future, but we do not have a date, correct? That is in the chat. Because I know that's a longer one, if you need to read it yourself, that you should be able to see that in the chat.
That is right. That, we want everybody to be able to participate, and we realize that if you've lost, some speech patterns, you won't be able to do that part of that study, but that's okay. You can still donate blood and CSF if that's what you wish, and that can be done remote. So the answer is yes, you can participate. I think to catch the end of that, I'm not reading it. We will be up and running, I think, before the end of the year, so sometime this fall. And we welcome, all folks.
All right, and that looks like it was the last question. Just a reminder to everybody that's still on, we are recording this, and we will try to send the recording out later this week to anybody who registered, so even those who weren't able to attend today. But I just want to thank the panel, Dr. Harris and his team. It was very informative, and thank you very much for presenting. And like I said, I'll get that recording out to everybody who registered later this week.
Take care, folks.
Thank you, everyone.